Well, good morning, good afternoon, or good evening to everyone, wherever you are. Welcome to another episode of DrugBank in Conversation. Today, we have the pleasure of sitting down with four individuals here from Genomate. My name is Chris, and I'm currently the scientific support lead here at DrugBank. I'll be the host and moderator of today's session. And today, we'll be discussing AI in precision oncology. A little bit about Genomate. Genomate helps oncologists determine the optimal targeted therapy for every cancer patient based on the individual molecular profile of their tumor. It's the world's first AI-powered clinical decision support system for precision oncology, transforming the lives of countless cancer patients. So just a quick note about today's format before we jump in. We'll be asking our guest today a series of questions for about 35 to 40 minutes. And then we'll open the floor for a live Q&A for about 10 to 15 minutes. And then we'll do some wrap up at the very end. If you have any questions throughout today's conversation, please feel free to add them to the chat bar or use the question function included on the platform. And we'll do our best to get to as many of them as we can once we open the floor for the live Q&A. So just before we start with the Q&A, the scheduled Q&A portion, I'd just like to quickly introduce our speakers. So to begin, we have Barbara Vodicska, Ph.D. She is the head of translational science, is a biochemical engineer specializing in prion diseases and cancer research. And as the head of translational science at Genomate Health, she plays a pivotal role in advancing precision oncology through innovative software development to improve targeted therapy decisions. We also have Robert Doczi, Ph.D., who is the head of research and a molecular biologist with vast experience in cellular signaling. And at Genomate Health, he spearheads the development and clinical validation of its groundbreaking computational method for precision oncology, leveraging cognitive computing to revolutionize treatment decisions. We have Istvan Petak, who has both an MD and PhD. He's the CEO, CSO, and founder of Genomate Health, and is a distinguished international expert in precision oncology and molecular pharmacology. He has over 25 years of experience, has pioneered molecular companion diagnostics and NGS in precision oncology, and his passion for innovation led to the development of Genomate, including the world's first proven AI-powered clinical decision support system for precision oncology. And then finally, we have Sean Khozin, MD, who is the Medical Affairs and Regulatory Strategy Advisor at Genomate, is a globally recognized oncologist, data scientist, and physician executive with extensive experience in therapeutic development and AI applications in biomedical research. And with a focus on cutting-edge innovations at the intersection of biology and AI, he brings a wealth of expertise to Genomate Health. So welcome to all of our guest speakers. Thank you for being here. Thank you for taking the time to join us. So we're going to jump right in to the panel discussion now. And to begin with, Istvan, could you please, just to make sure everybody's on the same page, give us a good definition of what exactly precision oncology is?
Thank you, and I warmly welcome all who attend this webinar. So we are discussing a very exciting subject and actually a dream of all cancer researchers and oncologists and patients when we talk about precision oncology. To really grab the essence of precision oncology, I always start with a little story and ask the audience what they would do in a situation when in a late night on call in an ER department, they see a patient coming in who complains about a terrible headache. And when they go closer, they see that there is a big nail in his head. So the big question is, what would be the appropriate therapy for this patient? And then I usually pause for a few seconds to let the audience think about this situation or imagine this situation. And then I relieve them by saying that, don't worry, we all know the answer. We treat the patient for migraine, because that's the most common cause of headache. We all know. So by protocol and based on statistics and statistical evidence, of patients who have headache, this symptom has a migraine as a disease and should be treated accordingly. So we have no protocol telling us what to do with the patient with a nail in the head. So it's just our reasoning and our understanding the functionality of the nerve system and the how the our sensory nervous system work. So we make a causal inference between the nail and the headache, and we create a hypothesis that maybe it's the nail. So our personalized precision therapy is to pull out the nail. So the same is precision oncology. In precision oncology, our dream is to treat cancers the same way. We do a very detailed molecular diagnostic test. We identify the nails. We know the nails now. So after the Human Genome Project, we systematically sequenced thousands of cancer genomes, and now we start to be able to say that we are getting close to the maximum number of cancer-causing genes, cancer genes, driver genes. The last counting or cancer genes, cancers counted 733 genes. This is according to Cosmic database. And we discover less and less. Actually, in 2020, there was a huge work done by an international consortium where they sequenced the whole genome of thousands of cancers and published in Nature the findings. And they published that they could identify at least one genetic driver in 95% of patients. So, and they identified that 2/3 of the driver genetic alterations were in the exome, the coding system, coding section, and there was also a 30% occurrence of driver alterations in the non-coding region. But actually, we started to see the cause of cancer. And it's so beautiful if you think about, you can take this driver genes and put it in a normal cell, and you can transform a normal cell into a cancerous cell. You see that they start to grow, they climb out from the petri dish, you can plant it in a mouse, and this grows. So actually, we now have a causative understanding. And so the question is, can we use this in clinical practice? Are we ready to use it? Or do we need statistical evidence for each? So right now, in clinical practice, we still use evidence-based medicine in our routine approach. So there is a broader meaning of precision oncology when we use a molecular biomarker to stratify treatments. But that's stratified medicine, not really the essence of precision oncology, because precision oncology, by theory, is an N of 1 therapy. So we make a personalized decision based on a functional understanding instead of a statistical decision. So when we use like a biomarker like EGFR or BRAF or HER2, these are the very known biomarkers, these have been approved as companion diagnostics of corresponding targeted therapies based on clinical trials which proved that patients who share the same biomarker have a higher chance to respond to targeted therapy. And this is why they have been approved, a statistically proven predictor of response. But but it's not really the essence of precision oncology. It's stratified medicine using molecular markers as an additional inclusion criteria or indication of a therapy. That's what's been always my dream, and many of us, and there will be several clinical trials which try to attempt to show that what if we just simply use our understanding of the molecular biology of cancer and we choose therapies according to this. And there have been several publications, including Shiva-01. This was the first trial. We will discuss it later, but also many others where molecular tumor boards assign individualized therapies. And So but the big question is how we will standardize this approach, because if it's an N of 1 therapy, we cannot be sure that we always think the same thing, because this is a headache with multiple nails, different sizes and different places in the head, and we don't know which one to pull out, and there is some debate on each case. So that's this problem which has to be solved, and this is a problem Genomate is trying to solve, but I don't want to tell the whole story to keep the others the excitement to share what's the solution for this problem. Thank you.
Yes, perfect. Thank you. Great introduction. So following on from that, I'd like to turn now to Robert and ask, in your opinion, why do we need AI in the field of precision oncology?
Thank you. So the need for appropriate variant interpretation has been addressed by the establishment of molecular tumor boards. They incorporate molecular biologists and geneticists into the clinical decision-making process. However, expert variant interpretation can be both expensive and time-consuming. So to facilitate the process, the current paradigm heavily relies on variant databases to classify the pathogenicity of individual alterations. As a result, several independent knowledge bases exist. And the existence of independent resources with different curators and scopes can lead to inconsistent or incomplete data collection. And besides inconsistencies in data processing, the underlying literature itself can also present inconsistent data due to the complex nature of biological systems. In addition, a key issue with the current practice of molecular profile interpretation, as it was already touched in Istvan's introduction, is its focus on finding actionable mutations or biomarkers associated with specific therapies, regardless of the full complexity of the profile. Needless to say, tumors don't exist because of single mutations, and just referring back to the pan-cancer analysis of whole genomes, it revealed that cancer genomes typically contain 4-5 driver mutations. As a consequence of these issues, of interpreting complex molecular profiles, treatment recommendations provided by molecular tumor boards show surprisingly low concordance. For example, there is one study which showed that in case of a really comprehensive molecular testing, there's just about 40% agreement rate between two well-established German molecular tumor boards. So more comprehensive tumor molecular profiles present greater challenges in interpretation. And on top of all of these issues, the problem of limited access to the expertise provided by molecular tumor boards in remote regions and non-academic centers is another challenge for implementing an informed decision-making approach. So all these issues can be addressed by an AI solution. Potentially, such an AI can compute genomic information in its entirety. It can aggregate various pieces of evidence. It can consider functional interactions. It can be readily accessible, and it can provide standardized, reproducible results for treatment decisions.
Okay, perfect. Yeah, and then just as a quick follow-up, how does Genomate specifically leverage some of these advantages of an AI-based approach in order to improve patient outcomes?
So I would summarize the key advantages of using an AI in precision oncology in reproducibility, standardization, and scalability, as in contrast to molecular tumor boards. Genomate provides a computational tool to address the previously mentioned challenges. But beyond the standardization, we also have clinical evidence that the system can contribute to improving outcomes. We have already published the reanalysis of data from the first precision oncology trial, which Istvan already has mentioned. It's called Shiva. And there, we could show that the system was clearly predictive of relative benefit of the drugs as used in the Shiva trial. More exactly, the scores assigned to individual treatments have predictive value for treatments that are highly likely to be effective and for those which have a high chance of failure. We also have lots of unpublished data in the pipeline based on both real-world and clinical trial data that confirm the safety and clinical benefit of the AI-supported clinical decision process. It has been implemented in molecular tumor board practice and provides meaningful assistance in therapy planning.
Okay. Yeah, perfect. Thank you. So I'd like to switch gears just a little bit here and turn to Barbara to ask how this idea of AI and precision oncology, and specifically how Genomate has implemented it, how does that actually work in a healthcare setting with healthcare professionals?
It's a good question. We have to clear off the concerns of healthcare professionals when we want to help adoption of these AI-based technologies into the clinical decision-making. So I would highlight two main things. One is explainability, and the second is clinical validation. So, but before all of these, for the adoption of this new technology, it is really important to understand that Genomate doesn't replace healthcare professionals, but it enhances their capabilities. So secondly, regarding explainability, it facilitates understanding and acceptance. So in contrast to many machine learning-based systems, Genomate uses a white box AI approach. This allows healthcare professionals to understand the logic on how the output was generated, and also they can browse through all the factors, the pieces of evidence contributing to the outputs. And this transparency fosters trust and encourages also a collaborative environment between clinicians and the AI system. And regarding the clinical validation, this is what really sets Genomate apart. It also increases the trustworthiness of the system. Our validation on the Shiva trial data already underscores the impact of our decision support. As Robert mentioned, we have several ongoing research projects which widen the applicability of the predictive value of the tool. And we are also committed to continually enhance the accuracy of our model based on emerging medical evidence and novel insights. And last but not least, I have to mention that, of course, users require regulatory compliance. They want to have their data safe in a securely operating, trustworthy system. So this is a key priority for us as well.
Yeah, definitely. And you mentioned regulatory aspects. So I'd just like to turn now quickly to Sean to see if you had thoughts about the regulatory side of all of this and what regulations might need to change in order for tools like this to be adopted in routine clinical care.
Sure, great question. I think the regulatory paradigm, both in the United States at the FDA and outside the US, including DMA and other ex-US authorities, international authorities, is evolving. And there's a lot of adaptation that's occurring. The adaptation is essentially pegged to these early use cases that these regulatory agencies are seeing. So there are changes that are organically part of this adaptation that we're going to see over the next couple of years. But I think what's important as each jurisdiction, every country and their regulatory authorities think about how to best ensure the safety and efficacy of AI-based solution, as we all adapt and regulatory authorities adapt, I think harmonization becomes quite critical. So rather than any specific changes that, I think we're moving in the right direction in terms of the regulatory innovations that need to occur, what's paramount is harmonization, because we don't want to be in a situation when there's discordance between how regulatory bodies in the US look at validation and assurance of safety and efficacy of these devices very differently than, let's say, the EMA, and that can cause duplicative work. It can cause a lot of confusion, and it may, in fact, become a bottleneck in advancing a lot of these tools, which can truly help patients. As we heard from Istvan, Robert, and Barbara, these are patient-centric solutions by nature, and they're really focused on advancing the health outcomes for the N of one. So any bottleneck that may exist in getting these solutions and putting them in the hands of clinicians so that they can help their patients really is not in the best interest of patients at the end of the day. So I think harmonization, regulatory harmonization, should be a key factor as regulatory authorities adapt. FDA has done a remarkable job in recent years, including the past few months. For example, FDA published 2 position papers on the use of AI and ML in advancing research goals and a lot of the precision oncology aspirations that the community has. So I think regulators are making all the right moves. It's going to take some time until a lot of these paradigms cement into concrete guidances. But as that happens, I think we need to ensure that we coordinate with regulators and coordinate among ourselves as an industry of precision oncology professionals to bring harmonization to the regulatory paradigm at the global level.
Mm-hmm. Certainly, yeah. So we've been kind of talking a little bit about, the practical applications of this in the clinical setting, the regulatory aspects of that. So I'd just like to ask, because I think a lot of people would be interested in a tool like what Genomate has to offer, but there's always hiccups and challenges to adoption. So I was wondering if you had any thoughts about ways that organizations can adopt tools like this, if there's any strategies they could use in order to make that transition smoother. So I'll ask Sean again, but then I'll also open it up to the other members of the panel, because I think this is really one of the key points is, again, putting the patient at the center of this discussion.
So I think one of the main factors in adoption is reimbursement. And payers also have to harmonize their thinking with that of regulators, because what regulators are essentially thinking about and requiring is clinical evidence that shows the safety and efficacy of these solutions. And that clinical evidence should ideally be sufficient to secure reimbursement. So we also need harmonization at the level of payers so that they can have the data that they need as these devices go through their regulatory schema to ensure and secure reimbursement. So I think that is a work in progress, and there's some room for improvement. But then beyond reimbursement, in order to increase adoption, physicians have to really think about and educate themselves, and it's incumbent on all of us to help educate each other on the proper use of these tools and how to incorporate them at the right time into the workflows, because computational reasoning, which is essentially what the Genomate AI provides, is a little different than sort of the type of reasoning that we do as oncologists, as clinicians, that's based on either average treatment effects, the aggregate experience of patients in clinical trials. And then we try to extrapolate from that aggregate average experience, which is a statistical concept, by the way, as Istvan mentioned. There's no average patient. That's a hypothetical statistical concept. So clinicians use their intuition to extrapolate the results of clinical studies to make individualized N-of-1 decisions. So that reasoning has to change, and it has to be able to accommodate computational reasoning so that clinicians can start to tailor the treatment decisions for the N-of-1. And that's a matter of education, but also trust. And Barbara, I think, hit on some of the main elements behind building trust. Explainability is one, and data, clinical data supporting the clinical validation of these tools. That's also not only important for regulatory decision making and reimbursement, but also it brings a lot of trust to the table. So one has to have a holistic approach that involves education and building trust in order to change existing workflows in clinical decision making, where we are using these tools to augment the intuitions of clinicians.
Definitely, yeah. Does anyone else want to add to what Sean was saying? I think this is a very important topic.
Just one notice regarding the acceptance and the breaking through regulatory and other hurdles. It is important to note that Genomate is a founding member of the Cancer Act Consortium, which aims to improve the acceptance and use of technological tools in oncology. It's part of the White House Cancer Moonshot program. So this hopefully will help for Genomate and other similar companies to gain acceptance in the daily clinical practice.
Okay, yeah, I think we can move on. But I think one thing that you mentioned, Robert, sort of leads really nicely into another question that we want to discuss, which was what sets Genomate apart from other solutions within the larger field of precision oncology, or maybe even precision medicine? And I think for that one, we can start with Istvan.
Thank you. So also going back to the previous question, the question is whether we can put the patient in the center. I can follow up on this. So the standard approach is that we are looking for patients for a drug, and not, but you should look for the best drug for the patient. So that's the difference. So so there are we have used single biomarkers as companion diagnostics to enrich patient populations who have a higher chance to respond, to find patients who have a higher chance to respond to therapy. But of course, now in the era of AI, many great companies are developing algorithms based on digital pathology images, for example, or simply based on X-ray images. Amazing. So machine learning can do amazing things. We can use machine learning to create an algorithm which predicts the response to a certain therapy. So we are replacing, these companies are replacing single biomarkers with algorithms, but they are still using the same paradigm. They are looking for a method to find patients for a drug. So that big difference, I think, which sets Genomate approach apart from the others, as what also Sean has mentioned, that we are using computational reasoning. And so our hypothesis was that similar to TMB, in TMB, there's a logical link between the tumor mutation burden, which means the number of antigenic alterations and the response to immunotherapies. We had this hypothesis that if we aggregate what we know about all the genetic alterations which are linked to the mechanism of action of targeted therapy, we can generate a scoring system like TMB, which will correlate to the chance or probability to respond to a certain therapy. So any targeted therapy which is associated at the highest level with the totality of genetic alterations of the same patient will have the highest chance to be effective. So this way we created a platform and a computational platform which actually can predict response to multiple therapies. And because if the same model is used to make predictions to multiple therapies for the same patient, logically, as a next step, we can compare them for the same patients. We can rank them and prioritize them for the same. So we can also support the decision of the oncologist to make a relative decision between multiple possible treatment options. So what we see more and more in clinical practice that there are many patients who have now multiple genetic alterations which are associated with an approved targeted therapy. As an example, we recently had a case of a breast cancer patient who had 7 pathogenic driver alteration. For example, she was a BRCA carrier. Of course, we know what to do with the BRCA. It's a biomarker associated with the use of PARP inhibitors. But the same patient had HER2 amplification and mutation. So, of course, we know what to do with HER2 positive patients. We give HER2 inhibitors. But the patient also had PI3 kinase mutation, and there is a therapy also associated with that. So the problem is that by the oncologist that provides the help of Genomate is to make a decision now which one to use, a PARP inhibitor, or HER2 inhibitor, or a PI3 kinase inhibitor. So to answer this question, what we need is a randomized clinical trial where we include patients with these 3 alterations, and then we randomize them into 3 arms according to this therapy to know which one to use. Now, I made some fun calculation how much time and effort we would need to do all the clinical trials we need to have evidence for which therapy to choose for each possible combinations of biomarkers. If we think that we have 733 cancer genes, as also Robert said, each cancer carries on average 4 or 5 alterations out of these, and each gene has hundreds of thousands of different types of mutations. So to cut it short, the end of the calculation was that we will need 14.4 million years to do all the clinical trials we need to have evidence which therapy to choose for each possible combination of genetic alterations. And even if we use real-world evidence, which is a solution, so not just to use data from clinical trials, but to collect outcome data of all patients, that can significantly accelerate the process. But I calculated still 100,000 years. So simply the sheer number. So we need a completely different solution, which eliminates the need of this phase 3 trial. So the Genomate solution is that we have a standardized algorithm which prioritizes treatments for each combination of mutations. Therefore, we can validate the algorithm and then generate evidence on the clinical benefit of the tool, the software tool. So that's the big paradigm. This is why when we use a new technology in medicine, there is a great acceleration of progress. So the progress is not continuous, there are big jumps. And we experienced a big jump when we invented the microscope and we could see the bacteria. Of course, it was a big change. And also when the molecular diagnostic methods were introduced, PCR and NGS. So there were like technological jumps. And why 2023 is very exciting is that, so we sometimes joke that there's a big hype around AI, but it's not just hype, it's really hope that really when you introduce to the system a completely new tool, a new technology, can make a very fundamental change, a paradigm change, because it's a new variable in the equation. So we changed the whole landscape by using a new technology. And that's why we are so excited that finally, with this tool like Genomate, and we hope that others will follow and we can compare who's the best. But for sure, we have been working on this for 8 years to develop a tool which really address the real essence of precision oncology, which is the N of 1 therapy decision. So to have a tool which chooses the best therapy for the patient and with a standardized way. And we think that having such tool can really transform how new targeted therapies can be used. We hope that we can accelerate drug development and also increase and accelerate the access to new targeted therapies, so we don't have to wait 14 million years while we have 20 people die every minute. So if you I don't want to make that calculation, how many people will die until we get the solution. So we hope that with technologies like Genomate, we can accelerate this process and save millions of lives.
Mm-hmm. Yes, thank you for that. That was an extremely in-depth answer. I think you touched on just about every aspect that we've talked about so far today. I'd just like to open it up to the other individuals if you had any thoughts on Genomate so far or the future, where you'd like to go next. We have maybe about another 4 minutes, and then we have a lot of questions from the audience, so I'll need to move on quickly. Maybe let's start with Barbara.
Yeah, thanks. I maybe would highlight some important benefits for HCPs and patients, what they can win from using Genomate and these new technologies. So first and foremost, using this informed decision making helps HCPs, since they get a ranked list of compounds based on predicted efficacy. This is totally new, and this enhances their confidence in selecting the most suitable treatment options, which is also good for the patients, so that they can receive the optimal, the most effective treatments early on, increasing their chances of a successful outcome. And of course, reducing these trial and error approaches, what is nowadays used by evidence-based medicine. Secondly, it also enhances time efficiency, not just by the quick software process, but also it can also save valuable time for healthcare professionals, because they don't need to wait for molecular tumor board sessions. They can refer to this tool and then administer the therapy. And also, this is also good for the patients, since they don't have to waste time on suboptimal therapies. And lastly, since this can standardize the clinical practice by introducing this method, Genomate can democratize access to cutting-edge medicine. Patients do not have to go to the best cancer centers. Since this being available online, Genomate can bring the latest medical advancement, the forefront of precision medicine, to every patient online, basically.
Yes. Sean, let's go to you next. Any final thoughts?
Sure. I think I want to actually underscore what Barbara just said about democratizing access to quality care. I think in a world where we're focusing on improving equitable access and equity in health outcomes, these tools are among the most viable way of providing access to quality care at scale. So I think that's something that I just wanted to underscore. And another way to think about how tools like Genomate's AI application raise the bar in treating patients is just to think about how we define precision medicine. Precision medicine is defined as giving the right therapy to the right patient at the right time and at the right dose. If we do that, mathematically speaking, 100% of patients should respond. If patients are not responding to therapy, then we have by definition, violated one of those variables. We're either giving the wrong drug to the patient or the right drug, but at the wrong time dose. So Genomate essentially overcomes that gap with computational reasoning and raises the bar for what we should expect from clinical decision making. That we, in any situation, if we treat 10 patients and only 4 of them respond to treatment, and we need to start to think about why the other patients are not responding. That's raising the bar and expectations that we should all have from individualized treatment and decision making.
Certainly, yeah. And then finally, before we go to audience questions, I'd just like to see, Robert, do you have any final thoughts you'd like to share?
Well, I think the others have pretty well summarized. Maybe just one aspect that could be also important is that the tool is agnostic of molecular tests. Obviously, the more molecular test results there are and the deeper they are, then the prediction will be, the better the prediction. But this not just agnostic, but also it can incorporate several tests in one run. So tests that were performed at different providers, and but then what? So the prediction is basically helping the clinician in the molecular aspect of the tumor. But then for the treatment decision, the final treatment decision is obviously in the clinician's hands, because there are other clinical aspects of a treatment. So it could be, comorbidities and toxicity and other drugs interactions and so on. So needless to say, I think, but just to make it clear that this is not intended to replace clinicians.
Mm-hmm. Yes, of course. There'll always be a need for that human touch in the clinical process, certainly. So this has been a really excellent talk. I feel like we could keep going for another hour, but unfortunately we need to move on. So we have quite a few questions here. I don't think we'll get to all of them, but we'll take them in the order in which they came in. So the first one is relating to the adoption of these AI-based technologies. How long do you think it would take before regulatory guidelines really catch up to the use of tools like Genomate?
So maybe I'll just give a quick answer to that. We touched on some of that. I think the regulatory paradigm is moving forward at a reasonable pace. I think the entities that should be catching up are payers, because there is a regulatory path forward right now for approving these therapies. However, there are cases where we have AI-based solutions, whether in imaging or other areas that have been approved by the FDA, as an example, but payers are not reimbursing. So I think we should really start to turn our focus onto payers and really start to think about when are they going to catch up. And there's a discrepancy here. When the FDA approves a cancer therapy, payers pay for it. It's a very much harmonized and aligned ecosystem. But when the FDA or other regulatory bodies approve or clear a software as a medical device solution, for example, that doesn't mean that the payers are going to provide reimbursement. So I think sure, there's some adaptation and evolution that regulators have to accommodate. But I think payers are much farther behind than regulators when it comes to this domain.
Yeah, and then another quick follow-up question that's a little bit more on the regulatory side, I suppose. To what extent is the Genomate solution aligned with NCCN guidelines?
Yeah, I'll just give a quick comment here. I think the guidelines of tomorrow are going to be algorithms, unless we stop making progress in biomedicine. if we continue to make progress in biomedicine at the current pace, it would be very hard for me to envision a scenario where in, let's say, 10 or 20 years, or the next generation of physicians are looking at documents in order to make treatment decisions. They'll be delivered those insights via algorithms as clinical decision support tools at the point of care. To me, that is where things are going, and I don't know exactly when that's going to happen at scale, but that is, I believe, where things are going. Currently, I think to me, NCCN guidelines and other guidelines, treatment guidelines, are extremely important. They're essentially the bare minimum that a clinician must know. To me, Genomate kind of goes a step beyond that, and are based on average statistical treatment effects. They're not based on N-of-1 studies. So the clinicians still have to extrapolate those insights to making individualized treatment decisions. And the body of evidence supporting these guidelines primarily comes from clinical trials. As we all know, clinical trials in oncology, the patients that are enrolled and have access to clinical trials can be very different than patients at the point of care. They tend to be younger, from more advantaged backgrounds, plus minorities enrolled in clinical trials. And as we all know, those of us that have done oncology clinical trials, we exclude essentially sick patients, patients with a poor performance status. And that's the body of evidence supporting these guidelines. So a patient with a very poor performance status that maybe is, comes from a disadvantaged background, a minority patient sitting in front of the oncologist, is not the patient that typically supports the clinical evidence that finds its way into guideline recommendations. So that's where Genomate essentially, again, raises the bar and goes beyond these generalized decision-making paradigms that are reflected in guidelines, which are important, but they're not necessarily about tailoring clinical decision-making.
Yeah, may I also add an example? Yeah, thank you. So it's very important, yes, as Sean pointed out, clarify that our tool is not a reference tool to learn about guidelines. But in clinical practice, as Sean said, today it can be used in combinations, because in guidelines now there are multiple treatment options. For example, I just got the great news about a lung cancer patient who has a complete remission now to nintedanib. It's a squamous cell carcinoma patient who actually is not a big surprise, although because the tumor had a FGFR gene amplification, and we know that nintedanib is an inhibitor of this, but FGFR amplification is not an approved biomarker of nintedanib, but nintedanib is approved as an approved drug to treat squamous cell lung cancer patients. Now, but for non-small cell lung cancer patients, in squamous cell patients, we have multiple treatment options, and we have immunotherapy as an option, and we have EGFR inhibitors. We have also nintedanib as an option, and several chemotherapies as an option. And we were just prioritizing treatment options for these patients, and we said that based on the molecular profile, which included, for example, an NRG translocation which activates EGFR, we said that maybe these patients should not be treated first immunotherapy and second nintedanib, maybe it's another order. It's just so we can personalize treatment decisions and stay within the guidelines. So that's the amazing thing, that because there are so many treatment options. And but of course, when we run out of approved treatment options and the and the physician is looking for the best molecular target-based off-label therapy, then also there are multiple options, and also there are multiple clinical trials, especially phase 1 clinical trials. And we know now that even if we direct the patient to the right phase 1 clinical trial, the patient might get benefit. So we have not thought about phase 1 trials this way, because we thought that they are just dose-finding safety trials. But actually, investigators MD Anderson showed that after 10 years of follow-up, patients who participated in molecular target-based phase 1 trials live significantly longer based on 10 years follow-up. So this shows the power of precision oncology and using what we already know. So that's the one of the idea of Genomate. Why don't we use what we know? And of course, this needs computational reasoning based on the level of complexity of the sheer complexity caused by the possible number of combination of these 4 or 5 genetic alterations. And so simply by using the power of the computer to make very complex mathematics, we can fight the complexity of cancer with the computer's power. It's just so simple. I mean, we know that already more than 20 years ago when the computer bet us in chess, and that Deep Blue, who won against Kasparov, was just a simple rule engine. It's nothing else. People maybe forget about this, but that was a very simple software. It's just simply the even that time the chip of the computer was just fast enough to compute all possible steps and win against the human brain. So imagine just this, that just using computational power, because now we know all the key chess figures in this chess. We are playing this chess against cancer, and there are 700 pieces in this chess game, and we have now 200 targeted therapies in clinical use and hundreds almost 1,000 in clinical trials. And these are our pieces. So what we have to know which one to choose, and we put forward maybe one, maybe in a combination, but still we want to win this battle and give chess, and at the end chess mate to cancer. And that's the beauty of using a computer as a tool. And I think what Sean is very right. These computational tools can reduce the dimensionality of the program tremendously, because now we see that as these guidelines are great, but they are getting to so complicated because all the comparative clinical trials, that's really hard to follow. So we need a dimensionality reduction. But still, I just want to emphasize that we believe in evidence-based medicine. It's just the subject of the device is different. So instead of using evidence-based medicines to develop drugs, we are using evidence-based medicine to develop a software tools. So it's just, otherwise it's the same. It's just we have to get used to the idea that software can be also a device and a tool to treat patients, like a phone endoscope or a drug or anything else. So this is the paradigm shift which has to occur to our thinking. So when we show like our clinical trial results and real-world evidence results, Robert was mentioning that Kaplan-Meier GURPS separate nicely, and where patients get Genomate AI supported therapy or a high-up score. If it were like a result of a new vaccine or new therapy, that would be really very clear. But when we say that this is just simply using clinical decision support software, a software tool, it's just amazing. And of course, we want to generate more and more data. And we want to make this available to the scientific community so they can use it when they interpret the results of such computational tool. But definitely with putting the software tool in the center of clinical investigations can really achieve a much better use of evidence-based medicine.
Certainly. There are always going to be things at which computers are simply better and faster than your average person. So I think that's actually an excellent place to end for today. So I'd like to, of course, thank all of our wonderful speakers for their time and for their willingness to come and have this conversation with us. I'd like to thank everybody watching, both live and after the fact, for your attention and your interest. This webinar is, of course, being recorded and will be shared with all attendees in the next couple of days. If you're interested in learning more about the Genomate team's work, you can visit their website at www.genomate.health. And of course, you can also follow them on LinkedIn. For more resources on the DrugBank side, you're of course welcome to follow DrugBank on LinkedIn as well, or visit drugbank.com. And in the meantime, stay tuned for future webinars just like this one. So thank you all once again, and I hope that wherever you are and whatever you're doing, that you have a great rest of your day.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.